And the mission is to work this source image to transform this source image to target image, and let's do apply two components of affine transformation. We can probably zoom a little bit because it's a little bit elongated, the patient's brain, and then also we can rotate it, so something like this, and so the position and size now matches between the two images, but definitely we are not satisfied. So what we need is some nonlinear component which simultaneously enlarge some structures. For example, the patient have much thinner corpus callosum right here, and I have thicker corpus callosum, and so the patient's corpus callosum should be enlarged, while patient's ventricle should be shrunken. So you have to enlarge a structure while shrink the other structure. So that's the nonlinear component. And the most popular way was developed a while ago, I don't know, like 2 decades ago, by SPM developers, I believe. And this is using a set of discrete cosine basis functions. So you use this linear combinations of discrete cosine basis functions to work the brain non-linearly. And so this is a cosine basis function, various cosine functions, and this bright color represent, let's say, shift the brain to the left, and the dark color shift the brain to the right. And if you combine, so these these inside matrices, they are like a multiplication of these two. So this one is multiplication of this and that, something like that. And you can apply this to the brain. This is of course 2D, but you can do this in 3D. So if you apply this to the brain, then you get this, for one part of the brain shifted left, the other part of the brain shifted right. And if you get 1,000 something like 1,000 linear combination of these functions, then you get pretty decent normalization. And SPM2 uses 1,176 combination and stop there. But is it enough? This is the SPM. Now we have SPM8, but SPM8 normalized button still uses this ancient module inherited from SPM2. So, so unless you use Dart2 or unified segmentation, this is basically nonlinear SPM normalization. And row A shows original images, 7 patient images, and row B shows the same individuals that went through FN only normalization, and row C shows the image after nonlinear SPM2 normalization. And it looks like that nonlinear normalization component here didn't do much to improve the normalization results. So, so the intermediate conclusion is that this nonlinear normalization based on discrete cosine basis function or polynomials, they all fail in dealing with severe diffuse injury. And the implication is quite severe. The upper row represents 8 control subjects' brain normalized by FN only and then nonlinear SPM2 normalization. And they look pretty much same, except a couple of them. But the patient's brain, the 8 patients' brain with severe diffuse injury, they look quite different. So if you conduct a study, case control group comparison study, and if you find a result saying that your activation blocks in patients are more larger in its extent, which is very frequently reported result in the imaging literature, then you don't know whether that's normalization failure or it's really the fact that patients have more varied location of activation focus.
Okay, so I'm here to ask the stupid question. So here it comes. Brace yourself. So here's what I don't understand, Hoon. You're studying patients who have all this atrophy, and so they have these big ventricles as a consequence of the atrophy. Somehow the warp is supposed, if it works right, they're supposed to now come out with brains with little skinny ventricles, right? What happens?
Right.
How do you then measure on what basis do you then measure the brain atrophy? Right? Are you not sort of taking it away in the warp?
So your question is how you quantify the atrophy?
Yeah, if after the warp you look at a brain no longer looks atrophied.
Right. So
then
so you should be happy about it. but your question goes beyond that registration issue, right?
I mean, presumably the reason for warping them would be so you could get some sort of measurement out, right, of the correlation of atrophy with behavior.
Right, so like a VBM analysis, say. Right, so
right. I think your question.
So if your warping is perfect, then that destroys your VBM analysis.
But it would be still useful for VLSM and for localizing lesions. Clearly, this is necessary.
Exactly, but I assume that in the case of here and with TBI or VBM applied to stroke or anything like that, right? You're interested in the atrophy. So why are you happy if it goes away in the warp?
It depends on your question. If your research question is that I need to align everyone's left superior frontal cortex. So if one patient's left superior frontal cortex is like peanut size, then I should blow it up and then make it as big as yours. And then if it gets as big as your superior frontal cortex, then I can run, statistic. I can ask the research question that whether that patient's superior frontal cortex activates more in this condition than yours. So if that's
That's
your
your question.
research question, then the closer the size gets,
Right.
Good.
the happier you will be.
Okay.
But if your research question is that whether that atrophy is related to that patient's impaired
And I
performance
was
in
just
conditioning,
assuming that was the question that you were interested in.
right,
so that,
right,
Yeah.
then you have to somehow quantify how much you bloated up that
see.
that patient's brain volume, and then that's exactly the next topic.
Okay.
So.
Okay.
So, the point of this slide is the same thing applies to the functional images too. So, it impacts on your functional MRI data too, not only the structural MRI. So we need a large deformation normalization scheme. So I mentioned that the SPM2 optimization protocol, it takes like 1,000 something parameters, but in large deformation normalization scheme, it goes to like millions. So, so this is the original brain with severe atrophy. This is affine only normalization. The third row is SPM2, and the fourth row, one of the large deformation normalization algorithm called symmetric normalization developed by Penn Pixel Lab, and the last row shows template, the standard template. Actually, it's custom built template, but you can see how powerfully symmetric normalization can warp the atrophied brain to make the brain look similar to the template brain. So to illustrate this normalization efficiency between different algorithms, I subtracted normalized standard template image from normalized image, and so basically this is intensity difference. So if registration went perfectly, then you will see nothing, and so the more white stuff and speckles you see, the registration went wrong. So not surprisingly, affine only worse than SPM2 and SPM5 unified segmentation normalization. SPM5 has this unified segmentation normalization scheme, which combines three spatial pre-processing into one: bias correction and spatial normalization, and what is the other one? Segmentation. Thank you, Dan. So that's the next. And SPM5 high dimensional warping algorithm was the big improvement, but again, large deformation normalization using diffeomorphism, which is symmetry normalization, performed the best. So there are actually several popular